Cloning has fascinated scientists for decades. From Dolly the sheep to cutting-edge genetic experiments, cloning has pushed the boundaries of what is possible. But as humanity prepares to expand beyond Earth, a new question emerges. Should we test animal cloning on the moon first? The idea may sound like science fiction, but it raises critical scientific and ethical questions. The moon offers a unique environment, one unlike anything on Earth. With 1/6 of Earth's gravity, no atmosphere and extreme radiation exposure, it is the perfect testing ground for biological resilience. Could cloning technology work under these conditions? Would a cloned embryo develop the same way in lunar gravity as it does on Earth? These are questions that scientists must answer before attempting to sustain life beyond our planet. Cloning is not just about making genetic copies of animals. It is about understanding how life itself functions at the cellular level. Every cell in a cloned organism must divide, grow, and form into a fully developed being. But what happens when this process occurs in an environment with weaker gravity and higher radiation? Would the genetic structure of a cloned animal remain stable on the moon? Or would space radiation introduce unexpected mutations? These are critical factors that could determine the future of space colonization. If humans plan to establish colonies beyond Earth, they will need sustainable food sources. Cloning livestock in space could provide a reliable way to produce meat and dairy without relying on Earth-based supplies. But before cloning is attempted on humans or livestock, scientists may test the process on smaller animals first. Mice, rabbits, and even amphibians could serve as the first test subjects for lunar cloning. These species have been used in cloning experiments on Earth, providing a valuable reference for comparison. The moon's harsh conditions would put every stage of the cloning process to the test. Would a cloned embryo implant successfully in an artificial womb under lunar gravity? Would its cells replicate at the same rate as they do on Earth? These are unknowns that must be explored before attempting human cloning in space. One of the biggest threats to cloning on the moon is radiation. Unlike Earth, which has a protective magnetic field, the moon is constantly bombarded by cosmic rays. These high-energy particles can damage DNA, leading to mutations or failed embryonic development. On Earth, cells have built-in repair mechanisms to fix minor DNA damage. But in space, where radiation exposure is significantly higher, those mechanisms may not be enough. Would cloned animals on the moon experience higher rates of genetic defects, or could they develop unique adaptations that allow them to survive better in space? Some scientists believe that testing cloning on the moon could reveal new insights into genetic resilience. If life can be cloned and sustained in lunar conditions, it could open the door for cloning in even harsher environments, such as Mars. Cloning is not just about replication, it is about the future of life beyond Earth. If we can successfully clone animals in space, we could one day use the technology to help preserve endangered species. Imagine cloning the DNA of extinct animals and bringing them back in space-based research labs. Would cloning become a tool for space-based conservation? Would it allow us to maintain genetic diversity in future off-world colonies? These possibilities raise both excitement and controversy. Ethical concerns surround cloning on Earth, and these debates would only intensify in space. Would cloned animals be treated as expendable test subjects in space experiments? Would they be able to live normal lives in artificial habitats? These moral questions must be addressed before cloning experiments move beyond Earth. Some argue that cloning should first be perfected on Earth before taking the risk in space. Others believe that the extreme environment of the moon provides the best test conditions to understand cloning under stress. If cloning works on the moon, it might work anywhere in the universe. But if it fails, it could expose vulnerabilities in our understanding of genetic science. One of the most ambitious ideas is using cloning as a way to seed life on other worlds. If we can transport genetic material to the moon and successfully clone living organisms, it could be the first step toward off-world ecosystems. Could cloned plants and animals one day thrive in self-sustaining lunar habitats? Would we use cloning to populate future Mars colonies with livestock and crops? These are questions that scientists are beginning to explore as cloning technology advances. NASA and private space agencies are already working on genetic research in microgravity. Stem cell experiments on the International Space Station have shown that cell growth behaves differently in space. Some cells divide faster, while others show signs of stress and mutation. What does this mean for cloning? Would an embryo develop faster in space or suffer from developmental issues? Lunar gravity is lower than Earth's, but it is not zero. Would this partial gravity be enough to allow normal biological development? Or would we need artificial gravity systems to support the growth of cloned animals? These are major scientific challenges that need to be solved. If cloning on the Moon is successful, it could revolutionize space travel. Long-term missions to Mars and beyond would no longer need to rely on frozen embryos or Earth-based supply chains. Instead, future astronauts could clone livestock, pets, or even service animals directly in space. Would space colonies have their own genetically engineered ecosystems? Would we see the rise of bioengineered animals designed specifically for off-world survival? These concepts sound futuristic, but they are becoming increasingly possible with modern technology. The ethical implications of cloning in space must be carefully considered. Would cloned animals on the Moon be the same as their Earth-born counterparts? Or would the extreme environment create irreversible changes in their physiology? Would future space-borne generations of animals evolve separately from their Earth-based relatives? Some scientists believe that space cloning could lead to rapid evolutionary changes. Without Earth's gravity, cloned animals might develop different bone structures, muscle mass, or even new biological traits. Could we accidentally create a new branch of evolution in space? Would cloned animals on the Moon develop adaptations that make them unrecognizable from their Earth ancestors? These are questions that only experiments can answer. If we can successfully clone animals on the Moon, we may one day take the next step, cloning humans for space travel. Would future space colonies rely on cloning technology to sustain their populations? Would human cloning in space lead to new genetic enhancements for surviving in harsh environments? These are questions that raise both excitement and ethical dilemmas. The potential for cloning beyond Earth is limitless, but it also comes with risks. The Moon provides the safest and closest environment to conduct these experiments before attempting them elsewhere. If cloning on the Moon works, it could pave the way for sustainable space exploration. If it fails, it could expose weaknesses in our understanding of biology and genetics. NASA and private companies are already planning genetic research projects that could lead to lunar cloning in the future. Will we see the first cloned animals on the Moon within our lifetime? Could cloning be the key to establishing permanent life beyond Earth? The answers lie in the hands of scientists and policymakers who will determine the ethical and scientific path forward. One thing is certain, the future of cloning is no longer confined to Earth. And as we look to the Moon as humanity's next frontier, we may find that cloning is not just a tool for science, but a necessity for survival in space. Would the Moon be the first place where life is reborn beyond Earth? The coming decades may provide the answer.